all of you for attending this evening, and thank you for the opportunity for me to present our work here. Just share the screen. So I'd like to briefly give you an overview of who I am. Many of you I have not met. Some of you I've met at various regional conferences, and some of the projects that we're concentrating on at the Indiana Center for Vascular Biology and Medicine with the support of the Cryptic Masons Medical Research Foundation. So I'm going to a brief biography because some of you don't know who I am. I'd like to briefly discuss our current projects and accomplishments and moonshots that we plan over the next decade. So I was born and raised in New York. I went to college and medical school at Columbia University in New York City. I did my general surgical training at the Massachusetts General Hospital and at the Brigham and Women's Hospital at Harvard Medical School. I took 3 years out to do a research fellowship in immunology at Harvard Medical School at that time, and then proceeded to a vascular surgery fellowship at Duke University in Durham, North Carolina. After a brief stint as a trauma vascular surgeon in Iraq, I took a faculty position at Indiana University School of Medicine in 2005, nearly 16 years ago. And I told my wife, who is from Boston, whom I met there, that we'd spend 2 or 3 years here and then move back to Northeast. But we both found that this was such a fertile area for my research and for running clinical trials because of the Midwest and the design of the school here and the how easy it is to raise a family. I became the Cryptic Masons Medical Research Foundation Professor in Vascular Biology Research in 2018. This is a very esteemed title. It means a lot with regards to academic influence when I apply for research grants or through the National Institutes of Health or through the Veterans Administration Center. Currently, I'm a professor of surgery. I'm an adjunct professor of cellular and integrative physiology. And with my work in immunology, I'm an adjunct professor of immunology. I serve on 3 National Institutes of Health study sections. I'm program director at the California Institute of Regenerative Medicine and a principal investigator for the Cardiovascular Cell Therapy Research Network at the National Heart and Lung and Blood Institute. So our areas of research and within the laboratory, we've been focusing on adult and induced pluripotent stem cells for regeneration of diseased tissue and interestingly modulating immune responses in chronic inflammation. And the diseases that we've been focusing on are peripheral arterial disease, aortic aneurysms, heart failure, wound healing, and with the current pandemic, COVID-19. So a part of peripheral arterial disease is atherosclerotic narrowing of the blood vessels that supply the leg. And what happens is that if there's insufficient blood flow to the feet, people develop breast pain, but even more concerning, there's loss of the skin and the subcutaneous tissue, what we call gangrene. And this is becoming a global pandemic before COVID-19 due to the increase in diabetes. Peripheral arterial disease in the United States at least afflicts over 8 million Americans. That's as many people who have coronary artery disease. However, the healthcare costs for peripheral arterial disease are twofold that of coronary artery disease. So it is a national healthcare concern. Critical limb ischemia is the end stage of peripheral arterial disease, as I mentioned, where patients do not have enough blood flow to support tissue nutrients. And we perform over 53,000 amputations each year in the United States due to critical limb ischemia. And 25% of patients will fail rehab after amputation. They can't use a prosthetic leg, and they end up spending the remainder of their lives in a nursing home. As a result, a lower extremity leg amputation is the 6th most expensive operation in the United States, costing over $10.6 billion a year. So way back when I was a fellow at Duke, I started to continue my research in cell-based therapies and was designed one of the first trials in the United States using a patient's own stem cells from their bone marrow to see if we could improve blood flow and prevent amputation. And this was a phase 3 mobile trial which was funded through the Cryptic Masons Medical Research Foundation. And very briefly, what we do is we aspirated bone marrow from the patient's iliac crest, like we would do for a bone marrow transplant. And with a partner, an industry partner here in Indiana, we had a closed centrifugation system, which was a really significant advance in the field. And it was placed in the centrifugation tube. And it separated out these stem cells where we could do it in the operating room in 20 minutes. And then we injected these cells into the lower leg. And this was at 26 centers in the United States. And what we found was at the end of 1 year, for those patients who were not diabetic, there was a significant reduction in major amputation. And that's an amputation above the ankle, so above knee or below knee amputation. And the p-value, which is what we as scientists look at, if it's less than 0.05, this was significant. And then when we looked at diabetic patients, there was a paradoxical increase in amputations in those that are treated with cells. So it became evident that the diabetic patient was a group that really didn't respond to cell therapy. However, when we looked at all of our patients in this multi-center trial, and 124 people, when we eliminated those with diabetics and with tissue loss, we found that there was a dramatic reduction in major amputation rates. And so what this suggested to us, this was evidence that, and as is evidence to us clinically, even with a surgical bypass, the best option to save a limb is that the diabetic patient was at greatest risk for losing their leg. They're also at greatest risk for having a heart attack or stroke or kidney failure. So it was that patient who had a foot ulcer, especially, that was at highest risk for amputation. So diabetes, because of the elevated blood sugars, leads to impairment of wound healing and affects the nerves and the nerves that innervate the muscles of the foot. And so there's pressure that's placed, especially on the first toe, right over the first toe prominence, what we call the metatarsal head. So 84% of amputations in diabetic patients are because of this diabetic foot ulcer. And so because I'm the beneficiary of the Cryptic Masons Medical Research Foundation, I can do a pilot study with that funding so that I can acquire data. And this is something I can do within a matter of 4 to 6 weeks because I need to get FDA approval and IRB approval. But with funding, we can do a pilot study of 10 patients, which will enable me to get data that will support an NIH or VA grant. So this was a gentleman. He was a 43-year-old veteran at our VA hospital in whom we injected mesenchymal stromal cells at the base of the ulcer. And this was actually 1 year later. Same patient, a different profile, a different patient with the classic Charcot's ulcer over the first metatarsal head. This was 24 weeks later, completely healed, saved foot. This was a young lady, diabetic, came to us from Louisville, who had this painful ulceration here on her toe. And this was 12 weeks later, completely healed, reepithelialized. So in this phase 1 pilot trial, it showed that 73% of diabetic foot ulcers are completely healed at 3 months after stem cell injection. And so what this enables us to do is in the next study that I plan to do, using a different cell population other than the patient's own bone marrow, I'll be using what's called mesenchymal stromal cells from a young healthy patient that will inject into the leg to prevent amputation. So our moonshot is to decrease amputations in patients with diabetes. And this is Dr. Stephen Miller. He's a colleague that's in our laboratory, and he has developed this, what we call the Tally-Ho genetic knockout mouse model. That's a diabetic mouse model, so it duplicates the human condition. And so what we're using are what are called spheroids. And these are aggregates of mesenchymal stromal cells from a 3D bioprinter that we purchased through a VA grant. We're also using what are called induced pluripotent stem cells that are where we take an adult stem cell and revert it back to an embryonic like stage. And then we put it in various growth factors to make it develop into a blood vessel cell. And I'm collaborating with an old friend of mine from Harvard, Dr. David Mooney, who's the chair of bioengineering at Harvard, who is using an alginate hydrogel. It's like a capsule, if you will, that And so we plan to inject these encapsulated cells. This is the capsule itself that contains the stem cells into the leg. And so these are some of the results. This is normal muscle from the tally-ho muscle model that we did not ligate or cause ischemia. This is a group that we created ischemia in, but we did not give spheroids. And this is the group that we gave spheroids to. And what you could see is between the untreated group and the treated group that was treated with the spheroids, there is profound regeneration of skeletal muscle. And this is key. It's key. It's very important in all elements of peripheral arterial disease. Patients who have mild disease called claudication, there's fibrosis of the tissue and impairs the ability to walk, but especially with critical limb ischemia, if we're able to regenerate muscle and blood vessels, we could save the life of the patient. And so there's a really, as I serve on various NIH study sections, there's a keen interest in using what the stem cells secrete. And so if we can encapsulate these cells, if you can imagine, you put them in a capsule, and they can secrete their products outside of that capsule, but they'll be protected because they're from another donor. They won't be cleared by the patient's immune system. Antibodies and immune cells can't attack these stem cells, but they can secrete the very important signals that lead to heart muscle regeneration or lower extremity skeletal muscle generation. And so this is an area that we are pursuing very aggressively. We have been selected as a core laboratory for the National Institutes of Health because we were able to purchase what's called the Buchi Encapsulator. This is a device that can create these alginate capsules in a process that we can use clinically. And so this is just a very brief picture that we took as we were developing our device. These are the capsules, these encapsulated stem cells, and this is a micrograph. And we purchased this Buchi Encapsulator through the Cryptic Masons Medical Research Foundation. So we now, because of your support, we are a core laboratory, a flagship laboratory for the National Institutes of Health for cell encapsulation. Another disease that we're looking at is aortic aneurysm. So in an aortic aneurysm, I'm a vascular surgeon, so this is a disease process very near and dear to my heart because I have to deal with this on an elective basis, but more concerningly, an emergent basis. But what it is, the inflammation, it's a chronic inflammatory process that seems to affect the aorta, which is the main blood vessel that comes out of the heart. And it involves the aorta just below the arteries that go to the kidneys. And this is where the arteries that they bisect, if you will, like a road that go to the legs. And this chronic inflammatory process leads to degeneration of the proteins and the cells, and it becomes weakened, and the aorta starts to enlarge. And when it reaches a critical threshold due to hemodynamic pressure, it ruptures. And that is a catastrophic event. A ruptured aortic aneurysm is the 13th leading cause of death in the United States. This is probably underdiagnosed because many people with ruptured aortic aneurysms die suddenly, and they're not diagnosed. They don't have an autopsy. And so it's probably much higher than the rate is much higher than this. And so when you look at the aortic aneurysm, when you look at in perspective of what we consider to be really significant diseases treated with surgery, the number of liver transplants each year in the United States are 7,000. The number of heart transplants each year are 2,000. But the number of aortic aneurysm repairs each year are over 45,000. So if you can imagine, if you will, you go into the doctor's office, you're somebody who's smoked, you're male, those are the predisposing risk factors. And your doctor said, well, let's get a abdominal ultrasound to screen for an aortic aneurysm. And so you're found to have an aneurysm of aorta. It's not large enough yet to repair, which is 5.5 centimeters. But you sit there and you think, oh my gosh, every abdominal pain I'm going to have, my back pain, is it going to rupture? Can I go on an airlines flight? Can I drive a car? It's like my patients tell me, it's like having a ticking time bomb in their belly. So a lot of the basic science research that we did in the laboratory through the VA system, looking at patients with and without aortic aneurysms, we found that there was a disruption in what we call anti-inflammatory regulatory T cells and pro-inflammatory effector T cells. So our thought was, if we can increase those regulatory T cells that throw water on the fire of inflammation, can we prevent aneurysm expansion by inducing what we call a type 1 regulatory cell, or for this discussion, a TR1 cell? So again, thanks to the Cryptic Masons, I was able to conduct studies in our mouse models of aortic aneurysm to get what we call proof of concept data. And so as a clinician and a scientist, but more as a clinician, I prefer to take our discoveries from the bench immediately to very safe and well-designed phase 1 and phase 2 clinical trials to treat our patients. And so we designed what's called the aortic aneurysm repression with mesenchymal stem cells, the ARREST-1 trial. And it was a randomized controlled trial where we looked at patients who had small aortic aneurysms. 36 patients, 12 got plasma-Lite-A, which is our placebo, and that's the solution that the cells are stored in. And then we had a low-dose group that received 1 million of these mesenchymal stromal cells, which were obtained from a young, healthy female donor. And this is 1 million MSCs per kilogram, 12. And then a high-dose group from the same donor who received 3 million MSCs per kilogram. And so we went from 1 year from a mouse study to outpatient treatment here at our VA, and these are our study coordinators. And we can only do this because we had the help of the Cryptic Masons. Otherwise, this would have taken 10 years to do. And so this is one of our veterans. He was the first gentleman to be treated. And so he's here in our ICU, received the stem cell infusion. And initially, our study, patients have to stay overnight to be observed because we're concerned about, the effects of the cells. But after treating 24 patients with no effects, it's an outpatient treatment that requires 30 minutes. And so these are our results. Prior to the COVID-19 shutdown, we had 28 of 36 patients enrolled. Our primary endpoint was, and it's called a surrogate endpoint, but I don't want to bore you, but it was the ratio of this anti-inflammatory TR1 cell to the pro-inflammatory Th17 cell at 14 days. And this is a profoundly significant p-value. And those patients that received the highest dose of mesenchymal stromal cells had the greatest response. And so we're going to finish enrollment in the study, but we're writing up the paper for the New England Journal of Medicine because this was this is an incredible discovery. And so we're designing also a phase 2 multi-center clinical trial in which we propose to use 2 doses of mesenchymal stromal cells. And this will be a more prolonged study that will look at the primary or the more clinically relevant endpoint in change in aortic size, which would be diameter and volume. And so a young resident in our laboratory, Catherine Leckie, presented this data at our International Vascular Surgery Symposium, and she received numerous awards for this. She also was an invited speaker, which is unheard of for somebody in their residency at the American Heart Association, their international conference in Philadelphia last year before COVID-19. Dr. Leckie was also developing a vaccine. And so this might get a little bit complicated, but there's a high association between aortic aneurysm and people who smoke cigarettes. And it has to deal with degradation of the proteins in the lung, notably what we call elastin. And as you can imagine, elastin is sort of like a rubber band. It expands and it contracts. And it allows us, the lung, to expand and contract with when we breathe in and when we exhale. The same thing with the aorta. The aorta is just sort of like your bicycle tire tube. It expands when the heart contracts, and then it contracts when the heart relaxes, and it sort of propagates blood distally. And so what happens is, our theory is, and this is an NIH grant, we just submitted a program grant, is that this elastin, when it becomes degraded, it becomes antigenic. It's just like with an autoimmune disease, like with lupus or type 1 diabetes. And what happens is these bad guys, the Th17 cells, attack the aorta, which is enriched with elastin. So if we can induce what we call immune tolerance to elastin, we can prevent the initiation of aortic aneurysms. And that's work that we're doing currently in the laboratory with our mouse model. So I'm going to shift gears a little bit, and it's going to go to heart failure. This is late breaking results. And this is from the Cardiovascular Cell Therapy Research Network, of which I'm a principal investigator, through which we conducted 4 clinical trials. But this is what we call CONCERT HF. And this was looking at patients who have what we call ischemic cardiomyopathy. And that's when we have hardening of the arteries in the heart, we have a heart attack, and we lose the heart cells, and we develop heart failure. So it's difficult for these patients to walk, climb stairs, to travel. So 6 million people in the United States have heart failure, and 50% of those will die within. So this study was looking at how we can treat patients with cardiac progenitor cells. And I'm trying to be as brief as possible. But this involved was we did a cardiac catheterization, and we took a snippet of heart tissue and we sent it to the University of Miami to Josh Hare's lab, and they separated out these cardiac progenitor cells. And then in the other group, I did a bone marrow aspiration, at which I aspirated bone marrow, and from the same patient, we collected mesenchymal stromal cells. So group 1 got the cardiac progenitor cells, group 2 got the mesenchymal stromal cells, group 3 got the cardiac progenitor cells plus the mesenchymal stromal cells, and group 4 got the placebo. And so what we did was we injected these cells into the heart. This is NOGA mapping. And NOGA mapping is when you put a catheter into the heart, you measure the electrical activity. And the lower the electrical activity means that that is an area that's at risk, the red zone, for heart failure. And so we use this, that's where we injected the cells. And so the results, which we just are now writing up, this was the first multi-center trial to assess a combination of 2 cell types from different heart tissues in heart failure patients. There was a significant reduction in those patients with adverse cardiac events, which is described as heart attack, heart failure, death, or stroke, in those patients that got the cardiac progenitor cells. And quality of life, that is the ability to walk, conduct your activities of daily living, was significantly improved by those patients that got the mesenchymal stromal cells alone and the combination of mesenchymal stromal cells and cardiac progenitor cells. And so we are submitting this report to the New England Journal of Medicine. It will be the second paper submitted to the New England Journal of Medicine because of Cryptic Mason's support. So COVID-19, as you all know, throughout the world, really has a significant impact. And what it involves, unfortunately, is this is the alveolus, and this is the working part of the lung itself. And this is the end terminus of the lung where air is exchanged. And so we get rid of carbon dioxide into the alveolus. That's when we exhale. When we inhale, we put oxygen into the blood where it's absorbed by the hemoglobin on red blood cells. Now with COVID infection, the COVID virus attacks what's called the spike protein. And this is sort of like a portal where it can inject its what we call ribonucleic acid, RNA. And what that does is it reprograms that poor type 2 alveolar cell, and it causes an inflammatory response, and it explodes. And that causes an inflammatory reaction where there's fluid buildup, there's cell death, and with fluid, you can't exchange carbon dioxide and oxygen, and our patients become hypoxic and require a ventilator. And so these just some images from a CT scan that shows infiltrates from a patient with COVID-19 infection. And this is a work that we did. So when we had to stop the arrest trial, but again, thanks to all of you, we had a stockpile of stem cells in our laboratory here at Indiana University, and we just changed hats and we got FDA approval on an emergent basis to treat our COVID-19 patients. So interleukin 6 is a bad messenger. It's like having a bill from the phone company, the water company, in your mailbox. Interleukin 10 is a good guy. It's like having, your tax return in your mailbox. But this is a patient that we treated. Interleukin 6 decreased, he survived. Interleukin 10 increased, this gentleman survived. This patient did not survive. He was already on a ventilator, failed mechanical ventilation. But interleukin 10, which is the good guy, increased. And this gentleman who survived, C-reactive protein, which is a bad signal, decreased profoundly. And so based on this data, and with my colleagues at the Cardiovascular Cell Therapy Research Network, about 3 or 4 months ago, we put forward a multi-center clinical trial which was awarded to assess the effect of multiple doses of mesenchymal stromal cells in treating patients with early respiratory distress. And the data that we were able to acquire, even though there were 20 centers in this grant, Indiana University, the Indiana Center for Vascular Biology Research, the Cryptic Mason's Medical Research Foundation, was pivotal in being able to provide proof of concept data to receive this award. And so our team here at Indiana University is very, very robust. We have many people that we work with. We have 4 study coordinators, various consultants, and we're working with colleagues at Stanford, University of Florida, University of Louisville, and Northwestern University. And finally, I would thank all of you for your attention, but most importantly, for your dedication to mankind and making the world a better place and enabling research. Thank you.
Thank you, Dr. Murphy, and thank you for